Hello dear friends, I Dr. Shamsher Singh welcome you all on my YouTube channel Pharma Learning. Today I am going to discuss about a very important topic, that is research, and it is a relationship to reverse pharmacology. So basically in the science you know that we know about pharmacology, clinical pharmacology, therapeutic pharmacology, but this is a novel topic which is called as a reverse pharmacology. So in this reverse pharmacology, I will just firstly discuss with you what is reverse pharmacology and what is its relation to research, why there is a need of reverse pharmacology in a research, what are the importance, or there are some limitations also. So basically if you will see, the word is reverse. Reverse indicate that you should back, and back to pharmacology. So it means the pharmacology, which is a branch of medical science, deals with the study about the drug. It deals with the study about all the mechanisms, all the therapeutic effects, dose, adverse effects, and therapeutic indications. So it means whenever we go for any drug discovery, there is a need that we should firstly know about the reverse pharmacology. So basically you can see here that whenever we go for a drug discovery, it take a lot of time, because as you know that any drug which is discovered, it minimally take approximate 13 years of time, with billions of dollars are required to keep one drug molecule from a lab and toward the clinical practice. It means if it take a lot of time and a lot of money, even though in the market, if your drug is unable to get success, if your drug is unable to get better effects or provide a better effects to the population, that mean sometime drug can be fail, sometime drug can be withdraw. In that case, there are lot of examples I can discuss with you, that there are so many drugs which are found to increase QT intervals. It means number of drugs which are known to interfere with the QT intervals. There are number of antibiotics. There are number of other anti-malarial drugs like chloroquine, and there are some drugs which increase the rate of repolarization. So many antihypertensives like sotalol and some other artemisinin drugs which are found to interfere with the heart, interfere with the contraction of the heart. It means whenever we synthesize, whenever we finally discover any drug in the market, if your drug will produce some type of adverse effect, and due to that adverse effect, if your drug is withdrawn, that means there is a scope for the research pharmacology, reverse pharmacology. So what is its reverse pharmacology? Suppose if you synthesize any molecule and afterward you are going for the preclinical analysis, and whenever drug will pass the preclinical analysis, that means pharmacokinetic analysis, pharmacodynamic analysis, toxicity profiling, like that, and afterward your drug is moved for the clinical trial and is available for the clinical practice. And even though in the clinical practice, if your drug produce many ADRs, and which due to that ADRs your drug will move back from the market, it means there is a loss of time as well as loss of money. So what it is, so what reverse pharmacology will do, it will do that the population data again will be analyzed on animal, and it will be again check on the clinically on the patients. So that mean you know that whenever there was availability of this sulfonamide containing ethyl glycol. There was availability of thalidomide. Even though after once availability in the market, they shown number of ADR, and due to that ADR, they were again mean analyzed on the animals, and they found that the toxicity was predicted. This means we are reversing back our pharmacology. So this is very important topic, that how drug discovery process is influenced by the reverse pharmacology. So basically this reverse pharmacology, I have discussed with you that how we can proceed our molecule. That means whenever we synthesize any drug which we called as a hit, and afterward it can become a lead molecule. So whenever any molecule X is synthesized in the lab, it is analyzed by two way. One will be the in vitro analysis, and after once we drive a dose, we go for the in vivo analysis in case of animals. Now, if in the animal your drug will successfully found to be beneficial, then we go for the clinical practice, mean we go for the clinical trial. Now, after once it cross the clinical trial and move in the clinical trial phase 4, then it means drug is allowed for the utilization in the population. Now, after the utilization, if you will found that there are some type of ADR which are reported by the pharmacovigilance, then this is a big challenge. So what you can do? You can do in case of reverse pharmacology, you see here, what you will firstly locate some target. That means target based drug design. So don't go that whenever any drug will be there, don't move that drug should firstly be analyzed by using some computer aided drug design or by using some techniques like crystallography. So it means whenever you will see in case of docking, we use some of the you can say receptor based docking or ligand based docking is there. So in that particularly we check that whether there is a better interaction of the drug with the receptor or not, or any ligand with the receptor or not. It means at this initial stage of synthesis we can do lot of changes. Instead we do the changes whenever our drug will be moving in the clinical trial phase 4. So this is a very good thing, that you should firstly identify the promising target protein, and this can be by the use of some in vitro or CAD like you can say study design. So this is a reverse pharmacology, that how we can go back and go for the drug analysis. So what were the major issues which were related to the reverse pharmacology? The basic issues were with the efficacy and safety. So it means in case of reverse pharmacology, if we easily identify some target, if we easily identify the drug interaction with that particular target, then it is easy that we can maintain the safety as well as efficacy of the drug. No, this is the history about the reverse pharmacology. It is well known that Sir Ramnath Chopra, he is renowned Indian pharmacology father, and afterward, this concept of reverse pharmacology was gained by a Sir Gannath Sen. He was a father of this reverse pharmacology. So he brought this topic, he used this term, that what is reverse pharmacology, how reverse pharmacology can be useful for the drug discovery process. Now, there are two things, that whenever we synthesize any molecule, we assume it that it can be a hit molecule. That means if it will be a hit, it will be having some binding capacity, it will be having some kind of structure, some kind of activity, and it can go for the lead. Now, the major thing is that whenever we found that our hit molecule, there are, because in hit there are number of millions of molecules, so hit will not be a single molecule, and out of million we get thousands. it can be a lead, and out of 1000 there can be a few which will be a active lead molecule. So that mean the lead simply indicate the molecule which is found to be having a activity on the receptor side. It can activate the receptor to produce some response. So but there is a need that we should optimize this particularly lead molecule. So for the optimization, we need some techniques, we need some in vitro techniques, we need some computer aided softwares. So these type of the technology is being, crystallography is being used, where we can go for the structural analysis. So that means if there will be any change in the, you can say any atomic change in the molecule can leads to change in the activity of that particular drug. So simply it can be little bit like a structural activity relationship. So this is that how hit molecule can move for a lead, and once the lead is optimized, then we can go for the in vitro in vivo analysis in case of animal. So what is the scope of this reverse pharmacology? Because it is having a lot of scope, that because whenever we go for drug discovery, there is a chances that our drug can be fail. So this reverse pharmacology can achieve a good scope, can provide a lot of, you can say, advantage objectives, in term that we successfully can move our molecule for the market and it can be utilized very widely. So this is the concept of reverse pharmacology, that this is a conventional technique where we are using any compound. This is basically a common pathway which we prefer from the previous time, that with the use of any molecule we go for the animal testing, then we go for the human testing in the clinical trial. But this is the concept of reverse pharmacology, that we should firstly know what are the drawbacks of the drug. So if we don't know, we should know that what are the drawbacks of these drugs which are related to that particular category. Suppose if I will say azole derivative, so azole can be ketoconazole, fluconazole, like that. So before going this, we should know that what are the drawbacks of the basic nuclei. So if we get this particularly the knowledge about the molecule, knowledge about the basic nuclei, knowledge about the particular category of the drug, then we go for the you can say animal testing again. That means we found that this, suppose this is a fluconazole, this is found to be having some adverse effect in the body, then we again check on the animal, then we should try to synthesize some novel molecule by taking the knowledge of that particular thing, and then we go for the, you can say, clinical analysis. So the concept of this reverse pharmacology is little bit different from the conventional method where we go for the drug analysis after the synthesis. No, what are the major domains of reverse pharmacology? Because there are three domains of reverse pharmacology. First is experimental, exploratory, and another is the experimental. That means hit, and then for lead, and then for the target based drug design. So what are the phases? First one is experimental phase, where it include a documentation of the clinical observation of biodynamic effect of standardized Ayurvedic preparation. You know that this reverse pharmacology gain a lot of, you can say, scope, because number of the herbal drug which nowadays are being preferred by the patients, it is due to the this availability of reverse pharmacology branch, and this is due to the safety of these particular herbal drugs. What is exploratory? Exploratory mean whenever we explore the drug analysis. Explore mean we should go from the lowest dose to highest dose. That mean we should check the tolerability of the drug in the patients. And the second thing, we should look for the drug interactions, that is there any kind of drug interactions or not. Then we go for the dose range finding. So this is very much essential, because due to this problem many of the drug they get failure in the clinical trial or after in the phase 4 of post marketing surveillance. The reason was, in that clinical trial only few doses were used. either low dose or high dose. But when we go for the clinical practice for use of drug in the market, then we should provide number of dose of the drug, and either we should provide that drug with combination of some other drugs. So this is the major drawback. That's why this exploratory phase is required. The third phase is again is the experimental phase. In this particular experimental phase, we should go for the sample to clinical analysis. That means this is little bit similar to this experimental phase. After once we explore the dose range of the drug, and after one we explore the drug interaction of the drug, likely these are the major properties. So what is the current scenario of this particularly reverse pharmacology? You know that in the old time, you know, or even though in the previous clinical trial, I have discussed that a huge time, approximately 13-14 years are required to keep a one molecule from a lab to clinical practice with availability of billions of dollars. That means lot of money is required. But with the availability of this particularly reverse pharmacology, now there is a less time is required to move a one molecule from a lab to clinical practice, and again it reduce the cost of analysis. How it reduce, I will discuss. This is a case study of artemisinin, which artemisia, this is used in a China particularly for the treatment of this fever as well as malaria against Plasmodium falciparum. And this is a well known example of this artemisinin, that it was found to become toxic, and in the reverse pharmacology number of the effects has been noted on, and the safety profile was analyzed. This is for the psoriasis. You know that basically I have discussed that a drug discovery process required 10 to 15 years, and with US dollar of 1 to 1.5 billions. So this is a basic process. But in case of this particularly reverse pharmacology, you know that within a short time we can analyze any drug, and because of less toxicity, and either cost could be more, but within a short time the drug will be available for the utilization. Next, in case of Parkinson, you know that mucuna pruriens, the seed of this plant mucuna was used for the treatment of this Parkinson, because you know that levodopa is already available as a primary therapeutic agent for the treatment of PD, not for the treatment for the management of the Parkinson disease, because it only slow down the symptoms. So how you can say this particular plant, which was available on the brand name of Zendopa, and it was approved by FDA. So this Zendopa had successfully, you can know that it is safe as compared to levodopa, because you know that levodopa, if it will be used for a long time, for 3 4 5 years, you know that there will be a drug tolerance, there will be a, you can say drug resistance will be there. So either there is a need of drug holidays, or either you can say you can achieve or use any other combination of the drug levodopa, carbidopa, and tacopone like that. So basically this was a one of the molecules zendopa which was available, and this was the benefit of reverse pharmacology. Now this for the osteoarthritis, you know that one of the project was completed a 5 year ago with expenditure of 2 millions, but its cost is just US dollar of 25 a month for a patient with a better therapeutic benefits. So that means ultimately this reverse pharmacology gain a lot of scope with availability of better, safe, efficacious molecule for the treatment of number of disorders. So this is the future scope, that if we use this type of the, you can say technology, or this type of the techniques where we use reverse pharmacology, and where we will be aware about the, you can say side or adverse effect of some of the drug, and it can easily be eliminated in a normal process of the drug discovery. So this is a future. Then I will conclude that this reverse pharmacology reduce the time of drug analysis, and reduce the toxicity, provide a safe drug, and reduce the amount which is required for the drug analysis, starting from the drug molecule synthesis up to the clinical trial analysis. Hope this topic was very useful to all of you, and this reverse pharmacology you can easily understand. You should like, subscribe, and watch these videos from the Pharma Learning channel. You are welcome, and thank you.